Now yesterday or in last class we completed this question. So we'll go to the 16th one now. Here, who was awarded Nobel Prize for synthesis of RNA in 1959? So here they are not asking about DNA, they are asking about RNA. So, who was awarded Nobel Prize for synthesis of RNA in 1957? Okay, it is yes, Oka, then A, Korenberg, C, Korana, then D, Nirenberg. So already these details in previous class I am given. So here the right answer is Oka. Yes, Oka is the right answer. So option A is the correct answer here. We are going to the next one. Now see, uridine monophosphate is found in. Uridine is nothing but uracil. Okay, so uracil is found in what already I told. Just find it out here. That is centrosome, lysosome, cell wall and RNA. So now tell me, it is nothing but the option D, that is RNA. I told you people. So in combination of DNA and RNA, DNA is containing ATGC combination. Okay, RNA is containing AGUC combination. So in this way, RNA is containing uridine monophosphate. So right answer is option D. We'll move to the next question. Now, Franklin and Wilkins showed that DNA, the four options are given here. DNA contains 4 bases, is a helix, has equal amount of adenine and thymine, okay, is made up of nucleotides. So, Franklin and Wilkins, they showed that the DNA is a helical structure. So, as a result, option B is the right answer. It is called helix. Okay, so we'll move to the next question now. Now see, the fusion reaction is a special test for fusion reaction. It is a DNA test. We'll go through the options here. RNA, DNA, protein, carbohydrate. The fusion reaction is a special test in case of DNA. So as a result, option B is the right answer here. We'll move to the next question. Now see, which one of the following is not a process of DNA replication? Which is not a process of DNA replication, DNA duplication, DNA formation? Okay, now we'll go through the options. Palindromic, dispersive, conservative and semi-conservative. So, DNA replication is including this dispersive, conservative, semi-conservative, but it is not palindromic. Now the question arises, what is this palindrome then? What is palindrome? So I am telling you, see, very simple. We are taking some of the names. Say for example, Malayalam. See the spelling, Malayalam. Okay, leave it. It is very big. We'll take very short name. Kanaka. We are calling it as Kanaka. Kanaka is one of the name. You read it from right to left or left to right. Read and see, then you will come to know, okay, both the side, you are pronouncing it as Kanaka itself. Why? Because the spellings are same. Okay? So such a type of duplication of DNA, whatever is there, in that DNA, the ATGC combination, whatever is there, okay, it is not same on both the sides. So as a result, it is not palindromic. So option A is the right answer here. We'll go to the next question. Now here, chemically DNA differs from RNA in having. Chemically, how DNA is differing from RNA? I told you ATGC or AGTC and AGUC. So thymine and uracil, they are changing in DNA and RNA. Thymine is found in DNA, but uracil is found in RNA. We'll go through the options here, then we will come to know. Thymine in place of uracil. B. Single strand. C. Ribose sugar instead of deoxyribose sugar. D. All of the above. So the right answer is option A. Thymine in place of uracil. Thymine and uracil, they are changing in DNA and RNA. Again I am repeating, in DNA thymine is present and in RNA uracil is present. So we'll move to the next question. Now see, the carrier of genetic information is. Who is the carrier of genetic information? So in heredity you studied, the parents, they are sending their characters to the offspring or the kids. In the sense, the parents are sending genes to the offspring. Genes are nothing but DNA. So we'll see the options here. First A, that is DNA. B, ribosomal RNA. C, ribosomes. D, transfer RNA. But here the right option is option A, that is DNA. DNA carries genetic material or a gene. We'll move to the next question now. DNA is found primarily. Where DNA is found? Okay, so I'm telling you, you know the cell. The cell is containing a big rounded ball inside, and that is called as the main switch of the cell. It is nothing but a nucleus. The nucleus, it is containing a elongated thread-like structure, what we are calling them as chromosomes. These chromosomes, they are containing the DNA material and it is called as gene. Many number of genes are present in that. Genes are also called alleles, allelomes, factor, trait, so on. So here the DNA is found primarily in cell nucleus, outside the cell nucleus, in cell cytoplasm, none of these. Okay, as I told, the nucleus is containing chromosomes, the chromosomes are containing genes, chromosomes are nothing but the DNA. So as a result, it is found inside the cell nucleus. So option A is the right answer. We'll move to the next question. Now here, the one which is capable of self-replication is self-replication. Here, the DNA is a material. It is forming its own kind of DNA. Okay, it is replicating, it is duplicating. So we'll see the options here. The one which is capable of self-replication: A, DNA, B, RNA, C, enzyme, D, protein. The self-replication is only found in case of DNA. So option A is the right answer, that we are calling it as DNA. We'll move to the next question. Now, replication of DNA is in. Where replication of DNA is seen? Okay, now see, 3' 5' direction, the helical structures, one is 3' opposite will be the 5', 5' where the another helix, if suppose one helix is there, one is 3' 5', another helix is there, okay, that will be opposite. If it is 3, if this becomes 5, if it is 5, this becomes 3. So opposite helixes will be there and they are quite. So here the helix direction they are given. Option A, 3' 5' direction. Option B, 2' 5' direction. Both 3' 5' and 5' to 3' direction. D, none of these. So here option C is the right answer. Both 3' to 5' and 5' to 3' direction. So in this way, the replication of DNA is taking place. So we'll move to the next question. Here, right option is option C. So as a result, so here we are stopping Unit 3, Part 1. The Part 1 is completed here. Then again, genetics in coming class we are continuing. We are continuing with Part 2. In Part 2, some of the interesting things are there. In the beginning, I am giving you the explanation, and after that, we are just going through some questions. So, we'll stop it here. Thank you one and all.